Thank you all for joining us today for our bioengineering seminar. It is my pleasure to introduce a bioengineering alum, one dear to my heart, and Anna Goestenkors. It's been too many years. And she got her bachelor's and her MN here from U of L. She's nice enough during her MN to be a GTA for me and help out in grading and so forth during this course when we got the class online and have the present in the room. And I got to know her well during that. She did a co-op with Dr. Magnuson, if I remember correctly, at Kaser, and then fell in love with her capstone project at Dr. T and devices. And so she did her MN with him and for PhD programs. And she works with Dr. Rutz, and is here today from Louis to tell us about her work. So thank you so much for coming into town. It's wonderful to have you back and doing all the great things you're doing.
All right, thank you for having me. I'm really excited to be here. And today I will be presenting on my PhD project, which is the fabrication of conducting polymer granular hydrogel bioencapsulating electrodes towards improved electrophysiological monitoring. So to start out, we see bioelectronics every single day in our lives, and we utilize them in many different ways. We see them in clinic in form of vagus nerve stimulators, pacemakers. We see them in everyday life in forms of glucose monitoring, and we also see them almost on every single one of us in the form of our smartwatches. In addition, we have begun using bioelectronics to monitor and or stimulate biological signals in a couple different applications, including in vitro models, robotic prosthetics, as well as within neurological interfaces. However, unfortunately, the application of bioelectronics in these fields, as well as many others, continues to be limited due to the fact that our traditional bioelectronic materials of metal and silicone fail to mimic our native cell and tissue environments. These materials lack three-dimensionality, porosity, hydration, and they are stiff, much stiffer on the gigapascal scale in comparison to the kilopascal scale of our cells and tissue. And this mismatch between the materials and the biological components that we're trying to interface with can lead to a variety of issues and limitations. When utilizing bioelectronics for in vitro monitoring and stimulation, you're interfacing with cells using a stiff planar material. And this can limit cell viability and proliferation. And even if cells are viable and do grow and develop, the way that they develop on these planar and stiff materials is much different than the way that they develop within our body. And so what we can learn from these devices and these monitoring and stimulation experiments is limited because it's not necessarily relevant to what's really going on. When utilizing the devices in vivo as implantables, these devices, because of that mechanical mismatch, can trigger adverse foreign body reactions. And essentially, this can cause the development of a fibrous capsule around the device that impedes the ability of the device to stimulate or monitor the biological components that it's interfacing with. And finally, when using these devices in vivo or in vitro, a common issue is that the surface area between the electrodes that you're using to monitor or stimulate and the biological components is limited. When you have topographically diverse tissue, as well as round and three-dimensional cellular components, the surface area that it interfaces is low, and this can lead to low signal quality and resolution, again, limiting what we can learn from these devices. However, researchers like myself have begun incorporating biomaterials, specifically hydrogels, at these bioelectronic interfaces to provide a more tissue mimetic interface for the biological components that they are monitoring or stimulating. A hydrogel is formed from a polymer solution, essentially polymer chains distributed in a large amount of water, and we introduce a cross-linking mechanism. And whether that cross-linking mechanism is a change in pH, the introduction of ionic species, a change in temperature, the goal is to bring those polymer chains together to form an insoluble polymer network. We can then swell this insoluble polymer network in a large water. Eventually, it contains so much water, greater than 90% by mass, that it becomes extremely hydrated. It possesses nanoporosity. You can use it in many different forms, including functional form, and they possess tissue-like softness, much closer to Pascal scale cells and tissues than our traditional bioelectronic materials. You're probably familiar with hydrogels in everyday life in the form of dermal fillers, you're wearing contact lenses, as well as different wound patches. A particular form of hydrogel is the granular hydrogel. The principal building block of this material is a micron scale hydrogel particle, typically between about 10 to 100 microns in diameter, and otherwise referred to as a microparticle. And when we densely pack these microparticles together, they form what is called the granular hydrogel. And this material, because of how those microparticles interact with each other, possess an interconnected microporous structure, as well as dynamic mechanical properties. And researchers seeing different granular materials and specifically granular hydrogels looked at this interconnected microporosity and dynamic mechanical properties and saw the possibility of using this material for many different things. They thought it could be handled very much like a solid in some states, but extruded via 3D printing or as injectables like a liquid, but maintain the 3D printed shape. They thought it could conform to different topographically diverse tissues, as well as encapsulate cells within the micropore structure. So they saw a lot of potential within the material. And in the biomaterials world, researchers have explored utilizing this material in various applications, including as a 3D cell encapsulation platform. So they're providing a much more tissue mimetic platform to culture these cells in growth and understanding. They've utilized it in 3D printing and 3D bioprinting. So different studies have been conducted where they have 3D printed different customizable structures. That's the beauty of 3D printing. You can print whatever you can. And then they seeded cells onto those structures, and the microporous structure within allows for the cells to infiltrate the bulk of the material. So again, you get a much more tissue mimetic material with cells throughout the bulk of it, but the customizability of 3D printing. On the other hand, some researchers have incorporated cells into the microporous space and then 3D printed the materials and cells simultaneously, so that, again, you have the customizability and efficiency of the 3D printing. The soft, hydrated nature of the material protects the encapsulated cells during the printing process. And then you have this nice, customized feature structure with a very consistent cell density throughout. And finally, researchers have used the material as an injectable towards tissue repair. So they can inject the material into different wound cavities, and because the material conforms to structures, it fills the entirety of the wound cavity, no matter what shape it is. And then over time, cells can infiltrate the microporous structure, as well as vessels can also infiltrate and help to regenerate the tissue of that wound in a way that's not achievable with bulk, non-nanoporous hydrogels. Simultaneously, researchers have been developing conducting polymer hydrogels, a different form of hydrogel. These hydrogels are formed from conducting polymer solutions, and these polymers have a conjugated backbone with alternating double and single bonds. And those alternating double and single bonds allow for, with a dopant, allow for electron transduction throughout that backbone. So it has electronic conductivity and can be used at bioelectronic interfaces to transduce signals back and forth, along with the advantages of being soft, hydrated, three-dimensional, and porous like traditional hydrogels. We have fabricated in the field various types of conducting polymer hydrogels, including this example of injectable hydrogels, and the Rex Lab specifically has worked in cast conducting polymer hydrogels, as well as 3D printed customizable PSS hydrogels. When looking at granular hydrogels, as well as the conducting polymer hydrogels, I started thinking, how can we combine these two types of materials to get the best of both worlds? Can we get the conductivity and can we get the functionality and versatility of these two materials by creating a conducting granular hydrogel? There have been attempts to make conducting granular hydrogels. They've been successful in the field. And there's two main approaches that have been utilized thus far, but they are very limited. So it's kind of, we're starting down the beginning of that path. The first method is fragmented conducting polymer hydrogels. So essentially researchers make millimeter scale conducting polymer hydrogels, like the previous slide that we just talked about, and then break those hydrogels up into micron scale particles. They've broken them up by extruding the hydrogels through smaller and smaller nozzles, or utilizing a homogenizer, which is basically a glorified science emulsion blender, to just break that hydrogel up over and over again until they get micron scale hydrogel particles. And the granular hydrogels formed through this fabrication method have great bioelectronic functionality. So when we're looking at conducting polymer hydrogels, we use a term called conductivity to evaluate how well the material will act at bioelectronic energy. And essentially, conductivity measures how well the material transduces electrons throughout the bulk of the material. So the higher the conductivity, the better it is to likely work at transducing signals back and forth. And this material, because they fabricated it from a pure conducting polymer hydrogel, has conductivity comparable to hydrogels already successfully used at bioelectronic. The ability to act in that manner is fantastic. Unfortunately, as you can see by the picture, that fragmentation method leads to a wide variety of shapes as well as sizes. And this can impact some of the functionality of the granular hydrogel that we talked about previously. Fragmented shapes can lead to less porous space, so you can have trouble encapsulating and growing cells in the material, and it can also impede extrusion. So clog nozzles and needles, or it can require a higher extrusion force, which can impact the accuracy of your print, as well as if you're encapsulating cells prior to printing, that higher extrusion force can be detrimental to cell viability. So it has fantastic bioelectronic functionality, but still some limitations for the end application. Another method that researchers have used is composite granular hydrogels. So essentially what they do with this method is they utilize non-conducting microparticle fabrication methods and non-conducting materials, but incorporate metallic or conducting polymer nanoparticles into the non-conducting hydrogen matrix. And this method generates highly uniform microparticles. You can get very low standard deviation. They're very circular. So those limitations we saw in the other method, this second method does not have those issues. However, you are incorporating the conducting components into a non-conducting hydrogel matrix. So the conductivity of these materials is typically much lower than the other method, as well as other conducting polymer hydrogel forms successfully in bioelectronics. So they both have their pros, but they both have limitations that we still need to work to overcome in the field. So for my PhD project, I decided that my goal was going to be to create a conducting granular hydrogel utilizing pure conducting polymer to achieve that higher conductivity and bioelectronic functionality, but also create a fabrication method that achieved the uniformity seen in the second method so that I could have the versatility of the end applications as we've seen in non-conducting forms of granular hydrogel. Within the REPS lab for conducting polymers, we typically use the conducting polymer PEDOT-PSS, polyethylene dioxyethionine polystyrene sulfonate. We utilize this conducting polymer because it has been widely explored in the field. It's commercially available. It has great aqueous stability and processability, so it just all around is really nice to work with. It has been used in the field to fabricate different forms of conducting polymer hydrogels, fast hydrogels, as well as 3D printed electrode configurations. And one of our most recent works that has been fabricated PEDOT-PSS scaffolds for in vitro culture. We've also explored the cytocompatibility of this material as well as other researchers and have found that it has great cytocompatibility. Past PEDOT-PSS hydrogels with fibroblasts seeded over top, and we saw fantastic proliferation and viability over multiple days. And as I said, it is commercially available. And most importantly, it's commercially available as an aqueous colloidal dispersion. And what does an aqueous colloidal dispersion look like? So essentially, you have nano and micro scale aggregates of positively charged PEDOT electrostatically attracted to negatively charged PSS distributed in a large amount of water. And what we know about this colloidal dispersion that I was particularly interested in and saw potential in was that elevating the temperature of this aqueous colloidal dispersion can bring those P.PSS aggregates together and cross-link them to form different P.PSS materials. Looking at this material and this cross-linking mechanism, I was immediately attracted to using a water and oil emulsion methodology. So when you think of emulsion, think of two immiscible liquids, like your salad dressing with your oil and vinegar. And when you shake that salad dressing, you get aqueous droplets of vinegar distributed in a large amount of oil, but they don't mix. The aqueous droplets are simply stabilized by the surrounding oil. And in a water and oil emulsion, biomaterials researchers have been using this to fabricate microparticles. And essentially, they add a polymer solution as the aqueous phase into the phase, and then introduce shear and turbulence to the mixture, usually with a stir bar or again a homogenizer. And that breaks that aqueous phase up into micron scale droplets stabilized within the oil. And then they introduce a cross-linking mechanism on the emulsion to stabilize those aqueous droplets into hydrogel microparticles. So my idea was, why not use P.PSS aqueous colloidal dispersion? It's a large amount of water as aqueous phase in a water and oil emulsion, add it to the oil, introduce a stir bar, break it up, and then raise the temperature to induce cross-linking of the P.PSS droplets to form P.PSS microparticles in the oil. And upon addition to the oil phase, P.PSS under shear turbulence did create these aqueous micron scale droplets within the oil. However, our next question is, what temperature do we use to cross-link these P.PSS droplets into P.PSS microparticles? There have been studies in the field on what exact temperature does to the aqueous colloidal dispersion, and research has found that temperatures between 60 degrees and 90 degrees Celsius induces behavior in that aqueous colloidal dispersion indicative of cross-linking. So for my study, I decided to conduct emulsions at 25 C room temperature as a control, as well as kind of the ends of those spectrums, 60 degrees Celsius and 90 degrees Celsius. And in the field, we can use what's called oscillatory rheology to evaluate the cross-linking of polymer solutions. So what you're seeing here is the P.PSS aqueous colloidal under 3 different temperatures. The gray trend line is called the loss modulus, and the black trend line is called the storage modulus. And in rheology, when the loss modulus is higher than the storage modulus, it's indicative that the material you're testing has a more viscous liquid-like nature, flowing more like a liquid. When those moduli reverse, it is indicative that the material is cross-linking and now is behaving in an elastic, solid-like nature. So as you can see, at 25 degrees Celsius, the aqueous colloidal dispersion did not see any change in the loss or storage modulus. It was not indicative of any cross-linking taking place at room temperature at all. At 60 degrees Celsius, we saw an increase and it almost crossed over, but not quite. And at 90 degrees Celsius, we saw a crossover between the moduli at 3 minutes. So very fast cross-linking there. So from this, we learned that the P.PSS aqueous colloidal dispersion does experience cross-linking at elevated temperatures, albeit a little bit slower, obviously, at 60 degrees Celsius. So I conducted 3 emulsion conditions then at 25, 60, and monitored the aqueous droplet formation over 60 minutes in these 3. And as you can see, at 15 minutes, the 90 degrees Celsius condition, those aqueous droplets get less transparent and much smaller, which we thought was indicative of cross-linking taking place in emulsion. However, we didn't see that change at 60 or 25 C. And indeed, when we measured the diameter of all 3 conditions, we saw a significant change at 90 degrees Celsius, but non-significant. We left it going for another till 60 minutes marks and at the droplet formation, and saw that finally at 60 minutes, the 60 degrees Celsius condition lost some transparency, not as much as we saw at 90 degrees Celsius, and got a little bit smaller. When measuring the 25 degrees Celsius diameter, we also saw a significant decrease, but we hypothesized at that point that that's most likely because of continued exposure to that shear turbulence than any cross-linking. At that point, we weren't seeing size or transparency visually. So at this point, we were left with what we hoped were microparticles distributed in a large amount of oil. So we had to go through a wash process to remove the oil and isolate the microparticles. To do so, you simply add the emulsion to centrifuge tubes, pack them down via centrifugation, aspirate out as much of the oil as you can, and replace it with phosphate buffer and safety, and repeat that process over and over and over again until you no longer see any large evidence of mineral oil within the microparticles. Mineral oil obviously would not be great for biological interfacing. So the goal is to get rid of as much as possible. And upon washing, we immediately saw that at 25 C, we added PBS, and the PDOT-PSS diffused into the surrounding liquid. So we did not get any stable microparticle generation at that temperature as expected. At 60 degrees Celsius and 90 degrees Celsius, though, we did see that we had stable microparticles at both of those temperatures. But you can probably tell that upon visual inspection, you can immediately see at 60 degrees Celsius that the size, shape, and transparency of those microparticles immediately looks very non-uniform, especially compared to 90 degrees Celsius condition. And when we measured the diameter and circularity of these different conditions, we did confirm that at 90 degrees Celsius, our microparticles were significantly smaller as well as significantly more circular. If you remember back, my goal is to get uniformity in these microparticles to be able to apply them to all these different functionalities. So from here on out, we decided to utilize 90 degrees Celsius fabrication conditions to fabricate our microparticles for our granular. And at this point, I was left with PDOT-PSS microparticles distributed in a large amount of PBS. So now the question was, how do we densely pack these to form the granular hydrogel? So I decided to utilize both centrifugation and vacuum filtration. In centrifugation, I simply centrifuge the microparticle suspension, they get packed to the bottom of the tube, and then I can carefully aspirate out the aqueous phase. Kind of like if you're doing cell culture, you pack a cell pellet down to the bottom and then aspirate out the media, very similar to that. Vacuum filtration, you simply pipette the microparticle solution onto a filter with about 1 to 10 micrometer sized pores, and the microparticles are isolated on the surface of the filter while the aqueous phase, majority of it is pulled out. And when that was complete, we were left with this paste-like material that we thought was a granular hydrogel, but of course we had to prove that it possessed those characteristic properties of the material. So first we wanted to look at the micropore structure. As you can see, PDOT-PSS is a very dark material, so you can't just look at it with bright field microscopy and be able to tell what's going on the inside. So I resuspended these microparticles in a fluorescent dye solution, dextran solution, and then repacked them using vacuum filtration with the hopes that any aqueous phase left, that fluorescent dye would only be left in the porous space of the material, and utilizing confocal microscopy, I would be able to fluoresce the void space of the. And as you can see, when an image using confocal microscopy, we were able to visualize the micropore structure within our material. The field uses void fraction to measure the porous space of granular hydrogels, and I measured a couple samples, and it's essentially the volume of the porous space divided by the volume of the overall sample, and ours pretty consistently runs between about 0.2 and 0.3, which is very close to what you see. And the pores are on average between about 5 to 25 microns in diameter, so very good if we want to encapsulate cells. Those pores are on the cell scale. However, at this point, trying to make a conducting granular hydrogel, I wanted to act at those bioelectronic interfaces, so I decided to measure the conductivity of the material. And upon measuring the conductivity, we got an average of around 7 Siemens per meter, which is on par with a lot of conducting hydrogels and even that conducting granular hydrogel fabricated through fragmentation. However, I got a 92% standard deviation, and over half of the samples didn't even produce a measurable conductivity value. So at this point, I had to figure out why my material was not possessing consistent conductivity. And I thought there were two possibilities. The first was that the packing of the microparticles was inconsistent. We want electron transduction not only within the microparticles, but between separate microparticles to transduce that electronic signal throughout the entirety of the material. However, I just told you I fluoresced these pores, and I looked at the connections and the number of microparticle junctions and the void fraction, and I wasn't seeing any significant differences between samples. So my next thought was that the conductivity in these specific microparticles was inconsistent. I'm seeing no difference in how the microparticles fit between samples. There must be something within the material itself. In the field and within my lab specifically, we have studied mechanisms for inducing more dense cross-linking of PDOT within the different PDOT PSS materials and improving the electronic properties. And one mechanism that we specifically have explored is the inclusion of ionic liquid. Now, the PDOT PSS aqueous colloidal dispersion is made with an excess of PSS primarily to impart aqueous stability, but the electrons are moving through the PDOT. PSS is not conductive. It's only there for that purpose. So if you leave too much PSS in the material and it disrupts those PDOT domains too much, that can cause inconsistent or low values. So including these IL or ionic liquid or IL to the middle aqueous charges on that ionic liquid come in and interact with the positively charged PDOT and the negatively charged stream. You separate them from each other so that your PDOT chains can come together more easily in a denser fashion and improve the conductivity of the materials. And we in the RUDS lab have explored ionic liquid in cast hydrogels. So we essentially mix neat ionic liquid into that colloidal dispersion, casted it into a silicone mold on glass, and then incubated the samples in an oven at 60 degrees Celsius for 17.5 hours. And we were able to formulate aqueous stable hydrogels. We evaluated these hydrogels for IL concentration effects on cross-linking density and the electrical properties as a result. We fabricated these hydrogels, soaked them in a lot of water to make sure they were at a state that could be used to interface with biological components, and then looked at how ionic liquid concentration affected the cross-linking of these hydrogels. And as you can see, increasing cross-linking density or increasing ionic liquid, we not only saw that there was a critical ionic liquid concentration, but we saw that as ionic liquid concentration increased, our hydrogels became smaller and smaller, which we thought was indicative of increased cross-linking density. You know, those chains are coming together more compactly and more dense within the material. An evaluation of hydrogel diameter with increasing ionic liquid concentration confirmed that we were seeing decreasing hydrogel diameter with concentration. And when evaluating these different hydrogels for conductivity, we saw that increasing the ionic liquid concentration 30 to 100 milligrams per mil produced a significant increase in the conductivity towards better bioelectronic interfacing. In the field as well, researchers have looked at solvent treatment as well as acid treatment of PSS thin films for improved conductivity. And again, what this is looking to do, acid or solvent solutions, they come in and they're looking to remove some of that PSS to allow the PDOT chains to come closer together. We recently looked at this mechanism in 3D printed PDOT PSS hydrogels. So these are hydrogels. We incubated these hydrogels in different solvent and acid solutions overnight, did vigorous washing to remove them, because again, we're trying to keep them good for biological change in diameter as well as a change in conductivity. And looking at the volume in comparison to the initially as fabricated hydrogel, we again saw that decrease in size with increasing treatment. Finally, with this form, we saw again a significant increase in conductivity, 2,000 siemens with these. So we know from the field and within our lab specific to PDOT-PSS hydrogel that this ionic liquid gelation agent and the solvent and acid treatments can help us form denser cross-linking networks in these materials and improve the conductivity. So for my microparticles, I decided to utilize ionic liquid as well as acetic acid within my fabrication method to try and generate better microparticles in terms of conductivity. So I added ionic liquid to my PDOT-PSS solution prior to adding it to the oil phase and creating these microparticles. We will now refer to them as PDOT-PSS-IL microparticles. And then afterwards, after they were out of the oil, I then resuspended them in an acetic acid solution overnight and then vigorous washing to create post-treated PDOT-PSS-IL microparticles. And both changes to the fabrication method produced aqueous stable microparticles. And when evaluating the diameter and circularity of these different populations, we didn't see any significant or detrimental effects by changing the fabrication process. So again, we're keeping that uniformity that we're looking for you have the versatility of the material. And when looking at the conductivity of the resultant granular hydrogels, I saw a significant increase with ionic liquid inclusion and another significant increase to about 150 here with acetic acid incubation. So I achieved greater conductivity. But I wanted to know what exactly was the mechanism behind this increase. Is it PSS removal? Are both of those things going in and doing exactly what we think they're going to do? So I looked at my microparticles and structured PDOT and PSS, and first utilized X-ray photoelectron spectroscopy to measure the amount of PSS in comparison to PDOT within the individual microparticles, and confirmed that with the inclusion of ionic liquid and with further post-treatment with acetic acid, we're getting a significant change from the original PDOT PSS particles in PSS content. So yes, we are pulling PSS out of those microparticles. In addition, looking at PDOT PSS, I told you PDOT's positively charged, PSS is negatively charged. And I thought, for pulling PSS, are we also changing the surface charge of the individual microparticles? So utilizing dynamic light scattering to measure the zeta potential of the microparticles, I confirmed that with PSS removal, surface charge of the individual microparticles becoming less and less negative. So we're able to tune the conductivity with these treatments and also tune the surface charge of the individual microparticle. Next, I told you that granular hydrogels possess dynamic mechanical properties. So I have all three of these microparticle populations now and three different types of granular hydrogels. So I wanted to confirm that these granular hydrogels possess the dynamic mechanical properties characteristic of the material. These mechanical properties are shear thinning, shear yielding, and self-healing. So in shear thinning materials, you will typically see with increasing shear rate applied to the material that the viscosity of the material decreases. And we confirmed that behavior in all three granular hydrogel populations. In shear yielding materials, the granular hydrogel under increased strain percent will start out at low strains with a higher storage modulus than loss modulus. A more solid elastic-like behavior, but as you increase the strain percent, those moduli will decrease and cross and continue, indicating that your material has yielded to that increasing strain and is now behaving in more of a liquid-like manner. And that was also confirmed in all three granular hydrogels. And finally, in self-healing materials and again at low strains at 0.5%, we see a storage modulus higher than the loss modulus, indicating solid elastic-like behavior. And when we move this strain up to 500%, those moduli reverse, indicating that it's transitioned to the liquid viscous behavior. But in a self-healing material, when you decrease the strain back to that low strain setting of 0.5%, the moduli recover their initial properties, meaning that the material can recover its different formations within it and different forces and return to its original solid-like state. And we confirmed that behavior over multiple cycles in all three granular hydrogels as well. When looking at the strain percent that the materials yielded at, there was no difference. And when looking at the amount of the storage modulus immediately recovered following high strain application, there was also no significant difference in granular hydrogels. We're seeing these dynamic mechanical properties are present in all three and no significant difference between the different populations. And it's these dynamic mechanical properties that lead to the ability to extrude the material from different nozzles and needles via it being extruded here. And what's happening is directly connected to those dynamic materials. When the material is being pushed through the syringe is applying greater and greater strain to the material. And that's causing those microparticles to move a greater and greater distances relative to one another, causing the viscosity to decrease. And eventually the strain becomes so great that the material yields to the increased forces and behaves in a more liquid-like manner and can be as a liquid phase. The self-healing property comes in when the material exits the nozzle and can be used to be structures. So here you can see it looks very much liquid in the video and the picture, but here we're able to print different three-dimensional structures that manner. And that's that self-healing property. Upon exiting the nozzle, the high strain that the material is experiencing is significantly reduced, and it recovers its mechanical properties and forces and junctions between the microparticles and can maintain the 3D printed structures in air. We confirmed that materials have, and we now have achieved good conductivity, we have dynamic mechanical properties, we have microporosity, and we're able to apply the material for 3D. And finally, I wanted to evaluate the material for cytocompatibility. So we utilized an ISO to evaluate the cytocompatibility of the microparticles, kind of acting in a medical device fashion. So for this ISO, you grow a monolayer of cells to about 80 to 90%, and then cover about 10% of the monolayer with your material for 24 hours, remove the material, and then evaluate the monolayers for changes in cell viability. And the benchmark is anything less than 30% reduction in cell viability is considered cytocompatible. Upon removal, we use fluorescent staining to stain our fibroblasts that I was using and image the different cell populations and quantify the cell viability. And not only did I meet the benchmark for cytocompatibility, we got greater than 98% cell viability in all three cell populations exposed to the microparticles. And there was no significant difference in cell viability in comparison to the control, which was just a monolayer of fibroblasts that didn't get exposed to any microparticles. At this point, we were looking to apply the material for a bioelectronic interface. And we formed a collaboration with different researchers at WashU and across the US in an NSF Frontiers grant. And they were looking to combine expertise in different insect olfactory systems, as well as robotics and biomaterials, which is where my lab comes in, to build different insect robotic machine brain interfaces. And the idea was that with the granular hydrogel that I created, we could use its conformability and 3D printability to create bio-encapsulating electrodes for studying olfactory systems in insects, specifically locusts. Insects sense their environment using their olfactory system in their antennas. Essentially, on an insect's antenna, they have multiple sensilla, and within that sensilla is an olfactory receptor neuron with the dendrites traveling up into the sensilla on the surface of the antenna. And when the insects are exposed to an odor, the odor molecules bind to receptor proteins in the sensilla, and that triggers the olfactory receptor neuron of that sensilla to form an action potential. And if the insect is exposed to enough odor, multiple action potentials along the antenna will form, and it generates a local field potential across the entirety of the antenna. Researchers have been studying olfactory systems in insects for years because they see how they can adapt to different and complex and dynamic environments and be able to sense the world around them much better than any of our electronic sensors that we make from traditional materials can sense the world around them. They utilize electroantenography to study how these insects detect odors on their antenna. There's a couple different setups. The top one you can see, they're essentially putting pin or wire electrodes in the antenna on either end and measuring the field potential change across the antenna when it's exposed to odors. That you can do intact with the insect. And then the second setup that you can do is encapsulate the ends of the antenna in glass capillary tubes filled with a salt solution and conductive wires to again measure the local field potential across the entirety of the antenna. However, researchers are fairly certain that the insects process the odor differently across different parts of the antenna, and that's what informs them about the direction and the concentration of the odor surrounding them. What they think is happening is that farther away from the odor, on the farthest point of the antenna from the odor, the action potential generated, the local field potential amplitude, is actually very low. And as you get closer on the antenna to the odor source, that action potential, field potential gets greater and greater. So what we wanted to do within this collaboration was use the granular hydrogel and the 3D printability to form precise bio-encapsulating electrodes around specific points of the antenna and measure how the local field potential amplitude across the antenna in response to different concentrations at those locations. So first, towards this goal, we wanted to look at, can this material perform in a basic electroantennogram setup? Can we encapsulate the ends of the antennas, interface those ends with sensing electrodes, connect those electrodes to our data acquisition system, apply odor, and be able to record signals generated as a result of those odors? I use thermal evaporation to fabricate custom mold electrodes. So you two sensing electrodes in the center, about 6 millimeters in diameter, and those are connected to contact pad electrodes at the end where we can use alligator clips to connect to the data acquisition system. I then deposited granular hydrogel deposits on equal sensing electrodes and placed X-size antennas into the granular hydrogels to where both ends are completely encapsulated by the material and still interfacing with the underlying traditional gold electrodes. And we were able to successfully record local field potentials generated across the antenna in response to odors. For every odor application, we have a red arrow, and every time we applied the odor, we were able to record local field potential. And when looking at the local field potentials of other electroantennograms using more traditional setups, our amplitudes were right on par with what the field is used to seeing. So the material successfully acts within this application as a bioelectronic interface, as a bioencapsulated electrode. So in conclusion, I have worked to fabricate different populations of P.PSS microparticles and form P.PSS conducting granular hydrogels with high uniformity for these end applications as well as high conductivity. We were able to showcase that the material has potential in 3D printing, has fantastic cytocompatibility for cell or biological interfacing, and fantastic bioelectronic functionality and can act at interfaces as a bioencapsulating electrode. In the future, we are looking to do more studies on the ability of the material to encapsulate cells as a 3D in vitro platform. I've done preliminary studies that show pretty good potential, but we're going to dive a little bit more deeply into that next. We also want to look at utilizing the material for 3D bioprinting cell encapsulating electrodes or cell laden electrodes, so we can precisely create multi-electrode arrays of cells encapsulated in a 3D environment and monitor them or stimulate them over time in a much more relevant environment. And also you can use the efficiency and accuracy of And finally, we are moving on to the next steps of creating this electroantenna multi-electrode array. We have ongoing studies that we're working to get to this point where we can start studying the differences in local field potential across the entirety of the antenna. I would like to take a second to acknowledge my lab, my PI, Dr. Alexander Russ, and specifically my undergraduate researchers, Justin and Cielo. They helped me with all of my microparticle fabrication, so without them, we wouldn't be able to get the results. I would like to acknowledge my funding sources. And I would like to take a moment to thank you all for inviting me up in this program. And a lot of what I'm able to do and achieve and the skills that I have is because of the skills and confidence and support that this program cultivates. So I'm always thankful for your guys' support, and it's a very big honor for me to present my work. So that's it. I can take questions now. So some conducting granular hydrogels that are not conducting, but just granular hydrogels in general that studies as in general. They typically will use materials that degrade over time. So a lot of times we'll connect it with a specific time frame in mind for staying stable enough to allow for cell and vessel infiltration over time, but then starting to slowly degrade and letting more and more cells and tissue regenerate and over time. And exit. In terms of my material though, as far as we are aware, it does not degrade at that time scale. And not seeing any application at this time, but it's something.
You're not starting. Gonna ask, 30% cytotoxicity is low as a cell person, not ideal.
Yep.
70%. I wondered your thoughts on that 30%, why you chose fiberglass. If the 50% is compared to cells that weren't in your hydrogel material or just cells that
Yeah, so when I was looking at the ISO, I was also surprised by the 30% reduction, especially when you're only applying the material to 10% of the monolayer. It's a low percentage, and so to see a 30% reduction. But I think the material is especially valuable for material leaching products. So I think that is a good part of the evaluation. So that's why we were interested in it. And of course, we were looking for a much less, we did not want that much reduction. We were looking for 90% or greater. That was the benchmark. So when we saw the 98% viability in all the different populations, we were very happy. The control is just for that ISO is just a monolayer of the cells not exposed to any biomedical device. Just incubated alongside them in the same environment for 24 hours without exposure. And I utilize fibroblasts. That's typically the cell that we work with most in the lab. Typically use collaborations to work with different other cells. I'm currently in a collaboration where we're trying to use the material to make follicles as a 3D cell culture platform, with the idea being to create a more tissue mimetic platform to allow the follicles to grow and the dynamic mechanical properties of the material to allow it to expand. So we're looking at different cell types, but that's not necessarily our area of expertise. So we look to collaborate with the university and outside so that we look to other. Oh, you asked the question that everybody in conducting polymers asks themselves every day, and everyone says, I don't know. I would say for our lab, we look at comparing to materials that have been successfully used in bioelectronic interfaces. There are thin films from conducting polymers that have conductivity up in the thousands of Siemens per meter. We have not achieved that level in our materials. It's definitely not in my material. But that's a different type of material. That's thin films versus hydrogels. And in the hydrogels, you have water, you're interfacing with biological components. So it's apples and oranges. But so we kind of look at it as what other research has been done in hydrogels that have successfully utilized it to monitor or stimulate electronic signals. And for us, that's around 10 to 100. We've successfully seen that in hydrogels. So my initial population of microparticle granular hydrogels technically met that threshold at 7, but it was just very inconsistent. So the inclusion of ionic liquid and further post-treatment, getting it up to about 150, definitely met that threshold and allowed for us to believe that it could be successful in bioelectronics. And then so that's we implemented it for the electron tentagrams and successful monitoring. Yeah, so we currently have, there's a PhD in our lab that is currently working along those lines to look at PSS. He's working in thin films now, but is trying to transition to hydrogels and looking at the ability to swell the hydrogel in different directions and use that to mechanical transduction for cell work. And so he's making thin films and looking at how swelling changes the conductivity and impedance of the material. So that's something that we're working on that's kind of coming up soon. We'll see.
Gels that like studied the same like size, you know, there's things for body. We probably say the same as you.
So I haven't looked at packing multiple millimeter sized gels. I would imagine that the properties would probably change. With the granular hydrogel, the background behind the different dynamic mechanical properties is that at the microparticle junctions, they are primarily dominated by frictional forces. And because the material is greater than 10 microns in diameter, but still on the micrometer scale, gravitational forces completely overtake thermal forces. So you get very specific mechanical interactions that lead to those dynamic mechanical properties. At the millimeter scale, I would guess that they would probably not operate in the same manner. Our original study on P.PSS IL hydrogels, that was on the millimeter scale. They were between around like 6 to 8 millimeters in diameter. And those we specifically looked at for cell interfacing. That was one of the first slides. It's a long way back. That we looked at seeing fibroblasts on those cast hydrogels. And we saw fantastic cell viability over multiple kPa. So these results are from our study on fibroblasts seeded onto cast P.PSS circular hydrogels for days, and we saw fantastic proliferation and cell viability. They became over good eventually. So the field and us have looked at that and saw great results, but I don't know how my material.
Any questions?
Look at contact resistance, start laying down those things on metal.
Yeah.
This is a difference.
Yeah, so we, in the future, the next steps for kind of interfacing and everything, start looking more specifically at impedance of the material, which is kind of the ability to transduce ionic signals to electronic signals for interfaces, but that's in the future. But there's also work in the lab creating kind of like a P.PSS adhesion layer on top of the metal electrodes that then interface with our materials, and that helps with the impedance and the electronic conductivity throughout the material. So we're working on it for future.
Any questions from anyone online? I don't think you have questions online.
Thank you.
Everyone has it with you?
I don't think so. Like, you only know a little bit with the P.PSS.
Yeah, we